Hello everyone. This recording covers mainly sections in Chapter 6, Sections 1 and 3. So let us begin with talking about enzymes. Enzymes are usually named after the reaction they catalyze, although there are some historical names that we still use. So most enzymes, not all, end in ACE. There is a precise classification scheme that groups the enzymes in a 4-level hierarchy and assigns each enzyme a unique number. And this number is based on function. So, for example, you could have malate dehydrogenase, for example, and have this enzyme from lots of different species, but it catalyzes the same reaction, so it would be given the same enzyme classification number. So, in order to talk about how reactions work and how enzymes speed up those reactions, we can start with a very sort of simple schematic diagram from our book in which we have reactants, this compound here, AB, where we're going to break the bond between A and B and form a new bond between B and C. So we have our reaction coordinate diagram. So you've seen these in your other chemistry courses, where the free energy is on the y-axis and the reaction coordinate is on the x-axis, and we start over here on the left with the reactants. And in order to go from reactants to products, we have to go through a transition state, and this delta G double dagger is the energy of the transition state. And you can see that the transition state, what's happening is the bond between A and B is being broken as the bond between B and C is being formed. So this is a very stable, very high energy intermediate, but it's something that we must do if we are going to get to the product where we have A and then the new bond between B and C. So the transition state is always going to be the point on the reaction coordinate diagram that has the highest energy. So the reactants must achieve a higher energy state called the transition state. And as illustrated in the previous slide, that's where bonds are broken and formed. And the structure of the transition state sort of leaves the structure of the reactant and approaches that of the product. And the activation energy is the energy required in order to attain the energy of the transition state. So what's interesting is the greater the activation energy, the slower the reaction rate. So that explains how we can speed up our reactions, that if we can somehow lower the activation energy, we can speed up the reaction. So before we continue though, let us look at different kinds of reactions and what you might encounter. So we might encounter a reaction where the reactants have a higher free energy than the products. And if we measure the change in the free energy of the reaction, that we'll see that we would have a negative delta G. Whereas we might have a reaction where the products have a greater free energy than the reactants, in which case our delta G would be positive. So if the delta G is negative, if the products have a lower free energy, we say that that reaction is exergonic. And if the products have a greater free energy, we say that the reaction is endergonic. Reactions may be one-step or multi-step. If enzymes are involved, they'll definitely be more than one step. So a one-step reaction has a single transition state, and a two-step reaction will proceed via an intermediate. So there will be two transition states. So the way that you tell is that the number of transition states will be equal to the number of steps, because every time you do a step, you have to go through a transition state. And the step with the greater activation energy is the slower of the two steps. And that's the bottleneck, or we refer to that as the rate limiting step, because the speed of that step will determine the speed of the entire reaction. So here is an example. So we can ask the question, which step is rate limiting, and is the reaction endergonic or exergonic? So this is an example of a reaction whose first step is rate determining, because if we look here at the free energy of the reactants, and we have to attain the free energy of this first transition state, and then we get to an intermediate, which is somewhat unstable, but it is still more stable than either of the transition states, then to get to this activation energy here, the energy we have to attain is not as great. So the first step is the slow step, or the rate limiting step. And again, if we compare the free energy of the reactants and the products, we can see that the products have a lower free energy. So this is an exergonic reaction, and it will have a negative delta G. So the role of the catalyst, as I hinted at before, is to decrease the activation energy. So, and this is the kind of graphic that is always used to describe this, which I do not like. And the reason I don't like it is because it implies that both the uncatalyzed and the catalyzed reaction proceed through one step, which is, for the catalyzed reaction, is never going to be the case. Maybe it is for the uncatalyzed, but definitely not for the catalyzed. But the point that is trying to be made here is that the catalyzed reaction will have a lower free energy, a lower activation energy, and its transition state energy is going to be lower. So one of the roles, there are many roles of the enzyme, but one of the roles is to stabilize the transition state. So in order to speed up the reactions the way they do, enzymes provide a multi-step pathway, again, which is not shown here, with several intermediates, and each one of the steps has a lower activation energy than any of the corresponding steps in the non-enzymatic reaction. So this is a little bit better. So let's go to an animation of this. This will load. So in this video, we show the red is the uncatalyzed reaction and the blue is the catalyzed reaction. So we can see, well, that was quick. Well, it doesn't look like I'm going to be able to show you that animation. Unfortunately, I'm having some technical difficulties, so I'll just walk you through it. So the red curve is the uncatalyzed reaction. So if you look at the red curve, we have one step, and we have to go from the energy of the reactants through this transition state to get to the, before we can get to the products. So this is the activation energy right here of the non-catalyzed reactions. That's what the N means. If you involve an enzyme, this shows three steps. So first the enzyme binds to the substrate to form the enzyme substrate complex, and it goes through that first transition state. Then we go through the second transition state, and then we form the enzyme product complex, and then the product dissociates. So there are three steps here, but all three of them have a much lower activation energy than that one-step reaction. So the enzyme catalyzed reaction is going to be much faster. So we can ask the question, how does the catalyst decrease the activation energy? Well, a number of ways. One is by binding the reactants, and this puts them in proximity to each other in the proper orientation and decreases their molecular motion. And then, as the substrate moves towards product, the transition state is stabilized. So let's talk about how this works. One of the ways we can talk about how this works is we can use an example just from organic chemistry that has nothing to do with enzymes. And we can look at a reaction where we have an ester right here and a carboxylic acid. And these can react to form an anhydride. So we can just measure the rate of that reaction and then just give it an arbitrary value of 1. Well, if you look at part A, you would see that you have two molecules, and in order for this reaction to occur, this oxygen was going to have to nucleophilically attack this carbonyl. So that is maybe somewhat difficult to do. You can speed this reaction up quite a bit if you have a molecule in which these are covalently attached. So this is just, you have your nucleophile and the carbonyl group in the same molecule. So you form this product right here, and this is 10,000 times faster. But we still have a way of maybe slowing these things down, because these are single bonds and they can all rotate. So what if I make this molecule right here, where I kind of freeze these things in place, and that way the carboxylate group, the oxygen on the carboxylate group is in a perfect position to nucleophilically attack that carbonyl, and I have a 10 to the 8th rate enhancement here to form the anhydride. So this illustrates how you can really speed up the reaction by making it very easy for the reactants to find each other and to be oriented properly for the reaction to occur. So reaction rates increase when reactants are brought into close proximity, when they're brought into the proper orientation, and the molecular motion is decreased. and a catalyst assists in all of these. So this is an old figure, but this shows sort of cartoony that if I have two molecules and the green patch and the yellow patch show that those are the functional groups that will be reacting, that a collision can occur if those functional groups are next to each other. This would be unproductive even if it's a high energy collision because the functional groups aren't properly oriented. So this is kind of like a goofy picture from our book, but it shows that if you have two reactants, they have a high degree of molecular motion, they have to collide with sufficient energy and in the proper orientation for them to react. And that could be difficult. But if I have an enzyme right here that's shown in this green blob over here on the right, the enzyme will collect these two substrates in its active site and slow down their molecular motion and bind them so that they are correctly oriented properly so their functional groups can be next to each other so they can react. So having an enzyme be able to accomplish this is going to greatly speed up the reaction. So that just describes how enzyme binds to substrate, but now we want the substrate to be converted to product, and in order to do that we have to go through the transition state as we just talked about. So we need to bring down the energy of the transition state to speed up the reaction. So that means stabilizing the transition state. So all enzymes are involved in transition state stabilization. In order to explain this, we are going to modify Fisher's lock and key hypothesis that we've talked about before. Where we envision that the enzyme binds loosely to the substrate but tightly to the transition state. And in so doing, that tight binding stabilizes the transition state and strains the substrate towards the transition state geometry and ultimately to the geometry of the product. So this is a way that we can illustrate this. Suppose I have a reaction and it's going to involve the breaking of a metal stick in two. So my substrate is envisioned to be a metal stick and I'm going to break it into two. And the transition state is a bent stick. So here's my substrate and I go through the activation of that energy that I have to spend to bend the stick, and then I get my product which has lower free energy because two small things have lower free energy than one big thing. So in this hypothetical reaction, there's no catalyst. Now here is what we envision if the enzyme were to be complementary to the substrate. So here, if my metal was a magnetic metal, and this is my enzyme, which is shown in this blue blob right here, and if these red things right here are magnets, and if this is a magnetic metal, you would see that you would form a very, very stable interaction between the enzyme and the substrate. So here's the substrate. I get a very, very stable enzyme substrate complex. But remember, my goal is to break the stick in two, and there is nothing here that's going to motivate me to do that. In fact, the only way I could break that stick is to pry it apart and then bend it and break it, and that actually takes more energy than without the so-called catalyst. So this is not going to work. So this is what works. If the enzyme instead is complementary to the transition state, the enzyme, which again is shown in blue, and these red are the magnets, the substrate binds loosely. So we have some nice interactions right here, kind of weak interactions at the end. But if I strain the stick towards the transition state, now my bent stick is stabilized, and that's going to help me convert my substrate to product. So what you see here in the blue is first my enzyme and my substrate bind to form the enzyme substrate complex, and then I form this transition state which is nice and stabilized by those magnets, and then I form the product. So in this reaction, I've substituted a one-step reaction for a two-step reaction, but I've stabilized the transition state, and that lowers the activation energy, and that speeds up the reaction. So our modified lock and key hypothesis then is that the enzyme is still the lock, but the transition state and not the substrate is the key. And the substrate structure is strained toward the transition state geometry. So we can say that the more tightly an enzyme binds to its reaction's transition state relative to the substrate, the greater the rate of the catalyzed reaction relative to the uncatalyzed reaction. So, or the greater the rate enhancement we get when we add the enzyme. So I don't really have a good picture of this, so I did my best here. So this actually describes the lock and key hypothesis really well, where you've got the substrate, here's our enzyme in yellow, and we form this really nice stable enzyme-substrate complex. But again, we saw that if you have a really nice enzyme-substrate complex, there's nothing to motivate you to form product. So this doesn't show this all that well, but imagine that this is my enzyme and the substrate has to strain towards the enzyme, the geometry in order to form the enzyme-substrate complex. So if I deform my substrate towards the geometry of their transition state, then the enzyme in the transition state bind tightly, and the transition state is stabilized, the activation energy is lower, and the reaction rate is faster. So, to summarize, how does an enzyme decrease the activation energy? Well, first, by binding the substrate or substrates and putting them into close proximity with the proper orientation of their functional groups that will be reacting, and in so doing, the molecular motion decreases. And once the substrate is bound, then the enzyme binds loosely to the substrate, but tightly to the transition state. That stabilizes the transition state, decreasing the overall energy, and by decreasing the activation energy, we can speed up the reaction. So how do we know this? How do we know that the enzyme binds the transition state tightly? Well, what we can do is we can do some experiments called transition state analogs, in which we can look for compounds that resemble the transition state, and those should be really powerful competitive inhibitors. So for example, if I imagine that this very simple isomerization reaction where I convert glyceraldehyde 3-phosphate to dihydroxyacetone phosphate, and say I propose that it goes through an enediolate transition state. Now, we can't isolate transition state. They're not around for very long, and they're very unstable. But I can make analogs. So this phosphoglycohydroxamate looks a lot like this. We've got the double bond here, and the geometry is the same, and the OH functional group is the same, and so is the phosphate. So if I can make a stable compound that looks like what I think the transition state looks like, then I can give my enzyme the substrate, which is glyceraldehyde 3-phosphate, or this transition state analog, the phosphoglycohydroxamate. And if the enzyme likes this better, then that means that I guessed right about what I thought the transition state looks like. It's like giving your enzyme either a stick or an already bent stick. The enzyme is going to go for the already bent stick because it binds tightly to the already bent stick, and it won't have to expend the energy to bend the stick. So this means that these transition state analogs are potent competitive inhibitors. This transition state analog will preferentially bind to the active site and exclude the substrate from binding. And it binds preferentially because the enzyme transition state association is much more stable and tighter than the enzyme substrate. All right, so now we want to form products. So we've got our enzyme and our substrate. We form the enzyme-substrate complex, but now we have to form product and we have to regenerate our enzyme. So the enzyme is going to do this too. So we are going to break bonds in the substrate and form new bonds in the product, and the enzyme is going to help us out there. So we're going to use the concepts we learned in organic chemistry, where we have nucleophiles, which are electron-rich, and electrophiles, which are electron-poor. And we should note the electrons in a bond stay with one atom and form an ionic species and a leaving group. And we will, as you did in organic chemistry, draw curved arrows from the nucleophile to the electron-deficient position of the electrophile. So what kinds of amino acids should we expect that have the ability to do this? Well, when we talked about protein folding, we said that proteins will fold so that the interior amino acids are hydrophobic, and mostly they are. But there are a few exceptions because we will need amino acid side chains that have the ability to act either as nucleophiles or electrophiles or as acid-base catalysis. And you can see that our top contenders, if we look at this chart right here, that we can see that histidine, aspartate, arginine, and glutamate, and lysine, cysteine, tyrosine are their percentage of catalytic residues is much greater than their representation of all residues. Whereas you see for glycine, glycine doesn't have the ability to do this. So we see more glycine in proteins, and glycine's not going to be generally part of a catalytic residue. So we want to introduce different terms so we can have something which we call acid-base catalysis, and we know that acids will use the Bronsted-Lowry definition. Acids are proton donors and bases are proton acceptors. So in acid-base catalysis, in the process of converting substrate to product, we will have amino acids that have side chains that can donate and or accept protons. And what that means is that these reactions are pH sensitive, because pH is going to influence whether these side chains are protonated or not. And in some cases, the side chain has to be protonated. In some cases, it has to not be protonated, depending on the mechanism. So the kinds of groups we expect to see are carboxylate groups, the imidazole groups on histidine, thiols, phenols, and those, of course, belong to these amino acids right here. So this is a figure from our book that shows the hydrogens in red. So these are all drawn in their protonated forms, but you can see that if any of those donated a proton, we would produce the conjugate base. And often in an enzyme mechanism, we need to donate a proton, and these side chains are capable of doing that. So again, if we look at this chart right here, again, look at the reactive groups and look at their charge at pH 7, and we can see that we can put these in groups in terms of principal functions. So just to highlight some of these, we'd imagine a carboxylate group that's normally negative charge will be able to be involved in proton transfer, but maybe cation binding too. We see histidine quite a lot, and that's because its pKr is typically near physiological pH. So a lot of times you'll see histidine in active sites because it's involved in proton transfer. When we get to looking at covalent catalysis, you'll see that cysteine and serine, if they lose their protons, they're are able to serve as nucleophiles, and when they do that, they form covalent bonds. So both of those are able to do that. So we'll have some examples to illustrate that. But before we do that, we should look at a pH rate profile. So pH rate profile shows the pH optimum. So reactions that involve proton transfer are pH sensitive because pH is going to influence the protonation of the amino acid side chains. And what we can do when we look at a pH rate profile is that we can look at the pKr values for the amino acid side chains involved in catalysis. So let me show you how we do that. So this is the pH rate profile for fumarase. We can see that at about 6.8, our rate is maximal. So if we measure the rate between, say, pH 4 and a little bit above 9, we can see that we have maximal activity at 6.8 and not so much around, say, pH 5 or pH 9. But the other thing that we can do is we can look at these inflection points. So I'll go back up. So the inflection points, so here the curve is going concave up to concave down, and here we're going concave down to concave up. So if we look at the inflection points, we can see that we have an inflection point about 5.5 and another inflection point maybe around 7.5, maybe it's a little higher. So this tells us it doesn't tell us which amino acid residues they are, but it does tell us the pKr. And if our pH optimum is 6.8, that means the pKr of this side chain, that means this residue, whatever it is, it's unprotonated because the pH optimum is 6.8 and the pH is higher than the pKr. This residue, again, whatever it is, has to be protonated because, again, the pKr of this side chain is higher than the pH optimum. So the reason this enzyme works well the way it does is because there are two amino acids. Again, we don't know which ones they are, but we do know it involves one amino acid that is unprotonated and one that's protonated. And the reason we don't work at the extremes is because down here, when the pH is really low, both of those residues would be protonated, and up here where the pH is high, both residues would be unprotonated and the enzyme won't work. So we can illustrate this by looking at ribonuclease. So ribonuclease, we've looked at this quite a lot. It's secreted from the pancreas during digestion. We've used this as a biochemical tool, but that's what Mother Nature made it for. Its pH optimum is about 6. There are 2 histidines. Remember, our ribonuclease is a single polypeptide chain, 124 amino acid residues. So histidine number 12 and histidine number 119, when the ribonuclease folds up there in the active site, those are the catalytic residues. And the pKr initial values, one of them has a pKr of 5.4 and one of them has a pKr of 6.4. So if I don't tell you which is which, you should be able to assign them based on the mechanism. So that's what I'm going to show you next. So this sort of brown fold right here shows us the active site, and here's histidine number 12. And notice that histidine does not have a proton. When we initially start this reaction, that proton is not there, whereas this histidine right here has a proton. So we can see why when we look at the mechanism. So here's our RNA. It's binding to the active site. It's shown in black. And what happens is this nitrogen right here acts as a base. And it pulls off this proton, so it accepts the proton. That means this oxygen right now is now an O-, and it's able to nucleophilically attack this phosphate. And we're going to, this is what we refer to as the scissile bond. So we're going to break this bond, and we can do that because the electrons in this bond will form a bond between oxygen and the hydrogen. So that means this histidine has to have a hydrogen because the hydrogen is going to be donated to the scissile bond. And what we do is we release our first product. So what we're doing is we're cutting this RNA. We're hydrolyzing it. So we released our first product. And again, this is a hydrolysis, so our second substrate is water. Now our water diffuses into the active site, so we have an intermediate here. So now this histidine is able to act as a base and accept a proton from the water. This makes water now hydroxide, which is a better nucleophile. It is able then to attack this phosphate, and now what we're going to do is we're going to break this bond, and these electrons will serve to bond to this hydrogen right here. So this hydrogen is going to be donated, so that means this histidine is now acting as an acid because it donates the proton. And to complete, we are able to, this is just a repeat of what's on the other slide. Now we're able to release our second product, and now we've regenerated the enzyme. It's not the same proton, it's a green one instead of a red one, but that's okay. We've regenerated our enzyme so the His 12 is unprotonated and the His 119 is protonated. So we can ask the question, does the enzyme use acid catalysis, base catalysis, covalent catalysis, or some combination? Well, we can see that we use acid-base catalysis because at various points along the mechanism, these histidines are donating protons, which means they're serving as acids, or they're accepting protons, which means they're acting as bases. They don't act as covalent catalysis. We'll get to that later, but covalent catalysis means that the enzyme and the substrate form a temporary covalent bond, and that didn't happen here. But now we should be able to assign the pKr values. So I will give you those values right here. So the histidine 12, its initial pKr is 5.4. It's less than the pH optimum, which we said was 6. So the pH optimum is 6 because 6 is greater than 5.4, so that means the histidine 12 was unprotonated. If you go back and look at the mechanism, that the histidine 12 was unprotonated. And that's the first step of the mechanism, is the histidine 12 acts as a base and accepts a proton. So it has to not have a proton to start with, otherwise it wouldn't be able to accept that proton. Whereas histidine 119 acts as an acid, so it has to have a proton to donate. So its pKr is higher, indicating a higher proton binding affinity, and also the pKr is greater than the pH optimum. So this histidine 119 starts out protonated. So the next thing we're going to do is let's do another one of those kinds of problems. So this is lysozyme. So lysozyme is shown here kind of in this slate blue color, and lysozyme hydrolyzes cell walls. And You might know about the cell wall structure from your biology classes, but the cell wall, it's a long polymer, but lysozyme just sort of takes a bite of the cell wall that has these sugars in it. So we are going to look at this mechanism, and ASP52 and GLU35 are the active site residues. Okay, so let's look at the lysozyme mechanism.
The lysozyme enzyme cleaves polysaccharide chains. First, the enzyme and substrate associate, forming an enzyme substrate complex. The enzyme catalyzes a hydrolysis reaction that cleaves the substrate into products, which are quickly released, allowing the enzyme to catalyze another reaction. The cleft in the enzyme holds 6 sugar residues of a polysaccharide. The hydrolysis reaction occurs between residues. Looking at the details of the reaction in solution, the sugar residues adopt their most stable three-dimensional conformation. However, after the polysaccharide enters into the enzyme substrate complex, the enzyme forces the sugar shown on the left into a strained conformation that more closely resembles the transition state of the reaction. Two amino acids within the enzyme facilitate the reaction. A glutamic acid donates a proton to the sugar on the right, and an aspartic acid attacks the C1 carbon atom of the sugar on the left. The attack on the C1 carbon results in a transient covalent bond between the sugar and the amino acid, and hydrolysis of the sugar-sugar bond. The deprotonated glutamic acid then polarizes a water molecule, drawing a proton away from it. This allows the water oxygen to attack the C1 carbon, breaking the sugar aspartate bond. In this way, the two amino acids are returned to their original states, forming the enzyme product complex. The enzyme and products dissociate.
So what we want to do now is assign the pKr values 4.5 and 5.9, and we notice here that the pH optimum is about 5. So that means that if the pH optimum is 5, whoever has the pKr of 4.5 is unprotonated. So that has to be the aspartate, because remember, the aspartate acted as a nucleophile. And that means that glutamate's pKr has to be 5.9 because its pKr is higher than the pH optimum, so it is protonated. And that corresponds with their mechanism. So we saw that the GLU35 donated a proton, and the ASP52 acted as a nucleophile, and it wouldn't be able to act as a nucleophile if it had its proton. And this is an example of not just acid-base catalysis, but also covalent catalysis, because a portion of the substrate was transiently covalently bound to the enzyme during the course of the mechanism. So I hope that this is helpful. Here are my references, and thank you for your attention.